Good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled Advancing Small Nucleic Acid Drug Development with a One-Stop Solution. And looks like Mark has joined us there already. Hi there, Mark. My name is Sonia Hunt, and it's my pleasure to be your X Talks moderator for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speaker. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit your questions and comments for our speaker throughout this presentation by using the questions chat box, and we'll try to attend to your questions during the Q&A session. Now, this chat box is located in the control panel, which is found on the right-hand side of your screen. If you require any assistance, please do not hesitate to contact me by sending me a message using this chat panel. I'd also like to welcome our attendees viewing this presentation from our LinkedIn Live event, and I encourage you also to submit your questions using the comments tab. At this time, all participants are in listen-only mode. Please note that this event is being recorded and will be made available for you, and have to register doing it on xtalks.com, okay? And at this point, I'd like to thank GenScript, who developed the content for this presentation. GenScript is a leading contract research organization in the world providing gene, peptide, protein, CRISPR, and antibody. Since its foundation in 2002, GenScript has grown exponentially through partnerships with scientists conducting fundamental life science research, translational biomedical research, and early-stage pharmaceutical development. The company is recognized as having built a best-in-class capacity and capability for biological research services, encompassing gene synthesis, peptide synthesis, custom antibody and protein engineering, and in vitro and in vivo pharmacology, all with the goal to make research easy. Now, it's my pleasure to introduce our speaker, and he is Dr. Mark Shulewitz, and as I said before, there he is. He earned a PhD from the University of California, Berkeley, and is an expert in molecular biology with publications in journals such as Oncogen. He has held key roles in the R&D and commercial teams of leading biotechnology companies. Mark possesses extensive experience with siRNA, particularly in developing cell-based assays for antibody discovery, development, and validation. He has successfully developed and launched numerous products for life science research. At GenScript, Mark brings his deep experience as a bench scientist to his role as a field application scientist, where he successfully provided technical consultation for multiple siRNA projects supporting their efficient progress. Now, with the introductions out of the way, I'm now going to pass the mic and the controls over to our speaker, Mark. So, Mark, when you're ready, you may begin.
Thank you, Sonia. Welcome to our webinar on advancing small nucleic acid drug development with a one-stop solution. I'm Mark Shulewitz, a field application scientist at GenScript, and I'm excited to guide you through today's session. We'll explore the fascinating world of small nucleic acid drugs, how they've revolutionized research and therapeutics, and how GenScript's one-stop solution can help you get started or advance your ongoing programs. Whether you're new to this field or a seasoned pro, I hope you find this webinar informative, insightful, and interesting. Let's dive in. First, let's start with an overview of today's webinar. We'll begin by answering the question, what are small nucleic acid drugs? Next, we'll look at the history of small nucleic acid drug development, including the drugs that are approved to treat human diseases. From there, we'll walk through a typical workflow, all the way from design to delivery. And finally, we'll round up today's webinar looking at some case studies that highlight these concepts. First, what are small nucleic acid drugs? To understand small nucleic acid drugs, it's essential to understand the role of proteins in human diseases. Proteins originate as genes encoded in DNA. These genes are transcribed into messenger RNA. In some cases, a pre-messenger RNA is formed, which contains introns that are removed by splicing. Messenger RNA is then translated into proteins, which exert their bioactivity on cells, or can also function extracellularly. Proteins have critical roles in human biology as well as in many human diseases. Some diseases are caused by the upregulation of a specific protein. In other cases, loss of a protein can lead to a particular disease. As a result, some therapies aim to treat disease by inhibiting overactive proteins, while other therapies treat disease by restoring normal protein function. There are three main approaches to inhibiting protein activity. First, there are small molecule drugs. Small molecules are synthetic chemicals that bind directly to proteins and directly inhibit their activity. Second, antibodies have been developed that, like small molecules, bind to proteins and also inhibit their activity. Antibodies can also engage the immune system, helping to eliminate cells that overexpress a specific protein on their surface that's recognized by the antibody, making antibodies particularly effective for treating certain types of cancers. However, it is difficult to deliver antibodies into the cell, so they're mainly limited to extracellular targets. Finally, there are small nucleic acid drugs, the topic of today's webinar. Small nucleic acid drugs are short DNA or RNA molecules. They're typically 15 to 60 nucleotides in length, and they bind to and regulate other biological molecules. The three most widely used types of small nucleic acid drugs, as shown in this figure, are aptamers, antisense oligos, and small interfering RNA. Aptamers bind directly to proteins and block their functions, much like antibodies and small molecules. Antisense oligos, also called ASOs, and small interfering RNA, also called siRNAs, target mRNA transcripts, preventing the translation of disease-causing proteins. Antisense oligos can also be used to restore normal protein function. Mutations in genes can result in non-functional proteins leading to disease. In some cases, antisense oligos can be used to modulate splicing to now generate an alternative messenger RNA that no longer contains the mutation and can be translated into a functional protein. Small nucleic acid drugs offer several advantages that make them attractive options as therapeutics. They can act on intracellular targets, they are relatively easy to design, and they can be synthesized quickly and easily compared to small molecules and antibodies. Let's look more closely at small nucleic acid drugs, starting with antisense oligos. Antisense oligos are single-stranded oligonucleotides that bind to RNA. They function in three ways. First is by promoting mRNA degradation, and they do this by directly binding to their target messenger RNA. In this case, antisense oligos are usually gapmers. That means that they have a DNA core that's flanked by the RNA. RNA directs the antisense oligo to its target messenger RNA, and then this forms a DNA-RNA hybrid between the DNA core of the antisense oligo and a portion of the messenger RNA. This DNA-RNA hybrid is now recognized by RNase H, which cleaves the target messenger RNA, leading to its degradation. A second way in which antisense oligos work is also by binding directly to RNA. However, instead of triggering degradation, these antisense oligos blocked the binding of ribosomes or initiation factors, thereby preventing translation of disease-causing proteins. Finally, some diseases are caused by mutations that create a premature stop codon or result in incorrect splicing that result in truncated inactive protein. And in some of these cases, antisense oligos can modulate splicing and restore a functional version of the protein. This is shown in more detail on the next slide. So in this example, a mutation in exon 2 creates a premature stop codon, resulting in a truncated version of a protein that is now non-functional. So to restore wild-type function, an antisense oligo is designed that binds to the region that contains the mutation, in this case, exon 2, such that following splicing, exon 2 is no longer in the messenger RNA. And this is called exon skipping. So now exon 1, instead of being joined to exon 2, is now joined to exon 3. And now this can restore protein function as long as translation through exon 1 into exon 3 remains in frame, and the protein that's created will now have an internal truncation due to the absence of exon 2. And despite this internal truncation, the protein must be functional in order for this to work. This situation only works in really specific cases, and we're going to address them. However, it's been successfully used to treat several human diseases, as we'll look at shortly. Now let's look at siRNA, or small interfering RNAs. So as we saw, antisense oligos can work by binding to messenger RNA and leading to their degradation through the activity of RNase H. Small interfering RNA, siRNA, can also degrade RNA, but through a very distinct mechanism. siRNA molecules are initially synthesized as a duplex. They're two complementary strands of RNA paired together. Upon entering the cell, siRNA associates with a protein complex known as RISC, the RNA-induced silencing complex. So once associated with RISC, one of the strands, the passenger strand, is removed. This strand is identical in sequence to the target mRNA and is not needed for silencing. The remaining strand, known as the guide strand, is complementary to the target messenger RNA sequence, and it directs the RISC complex to recognize and bind to this target sequence. Once bound, the RISC complex now catalyzes the degradation of the target messenger RNA, effectively silencing gene expression and reducing protein levels. siRNA is based on microRNAs, miRNAs, which are naturally occurring non-coding RNAs found in eukaryotic cells that regulate gene expression. MicroRNAs are transcribed as a premature form called a primary microRNA. Primary microRNAs form a characteristic hairpin loop structure that are flanked by 5' and 3' sequences. During microRNA biogenesis, primary microRNAs are processed by DRASA, an enzyme that removes the flanking 5' and 3' sequences, generating pre-microRNAs. The pre-microRNAs are then exported to the cytoplasm, where they undergo further processing by dicer, an enzyme that removes the loop structure, leaving behind the mature microRNA. The mature microRNA is now loaded onto the RISC complex. The passenger strand is discarded, and the guide strand guides RISC to the target messenger RNA, which is now degraded, leading to gene silencing. Mature microRNAs and siRNAs share a very similar structure, but there are some key differences. So first of all, siRNAs are designed specifically to target genes for which no naturally occurring microRNA has been identified. And furthermore, siRNAs have perfect complementarity with their target messenger RNA sequences, leading to precise and efficient degradation. MicroRNAs, in contrast, typically have mismatches with their target mRNAs. So the different intermediates in microRNA biogenesis, the primary and pre-microRNA, may look familiar because they serve as the basis for another class of gene silencing molecules involved in RNA interference, small hairpin RNAs, or shRNAs. The traditional shRNAs resemble pre-microRNAs and are transcribed from DNA. Once they're expressed, they enter the RNA interference pathway and are processed by dicer, which removes the loop to produce the functional siRNA. There's another form of shRNA, shRNA mirs, and these are identical to shRNAs, except they contain the 5' and the 3' flanking regions. And once they are produced by being transcribed from plasmid or viral DNA, because they contain the 5' and 3' flanking sequences, like primary microRNA, they enter the RNA interference pathway at the level of DRASA, which processes them by cleaving off the 5' and the 3' region, converting the shRNA mir to now an shRNA that is then processed by dicer and then loaded onto the RISC complex, and so on. So although shRNAs are widely used for gene knockdown, they are not considered small nucleic acid drugs. However, I wanted to introduce them because they play an important part in research and functional genomics. So for the rest of this webinar, we'll focus on siRNA, as it is one of the key small nucleic acid drugs used in therapeutic applications. Now that we've introduced small nucleic acid drugs, let's take a quick look at the history of small nucleic acid drug development. This figure shows the history of small nucleic acid drug development, starting in 1956 with the development of RNA hybridization and extending all the way to modern day. Some of the important advances along the way include, in 1984, the development of chemical modifications to antisense oligos, the discovery of microRNA in 1993, and the discovery of RNA interference in 1998. And these last two discoveries, microRNA and RNA interference, were awarded the Nobel Prize in Physiology or Medicine in 2006 for RNA interference and 2024 for microRNAs. And the awarding of the Nobel Prizes illustrates the importance of this discovery, not only to understanding human biology, but also to their application to basic research as well as to therapeutic development. So the first small nucleic acid drug to be approved for human use was Vitruvine in 1998. It was an antisense oligo, and it was approved for treating CMV, cytomegalovirus infection. To date, 11 antisense oligonucleotide drugs have been approved. They are shown in orange in this slide. In 2004, the first aptamer drug was approved. This was Macogen, and it was approved for treating age-related macular degeneration. To date, only 2 aptamer drugs have been approved, the first one in 2004 and the second one in 2023, and they are shown in blue. And then finally, the first siRNA drug was approved in 2018 Onpattro, and it was approved to treat neuropathy associated with hereditary transthyretin amyloidosis, or HATTR. Even though siRNA's approval lagged behind antisense by about 20 years, they are quickly catching up. To date, there have been 8 siRNA drugs approved for human use. They're shown in green in the slide, and you can see that there have been a lot of approvals in the last few years. The table organizes small nucleic acid drugs based on their type, the mechanism, and the target. So the first 2 aptamer drugs are on the top. Both function by binding and inhibiting their protein target, much like a small molecule drug. Next down the table, we have antisense oligos. So Vitruvine, the first antisense oligo to be approved, functions by blocking translation. However, it's the only one that functions by this mechanism. There are 5 other antisense oligos that function by splice modulation, and then 5 additional ones that function by mRNA degradation. Looking at the splice modulators, you can see that they actually target 2 different genes, and in fact 4 of them target the same gene, DMD, which encodes dystrophin, and mutations, loss of function mutation in dystrophy lead to a progressive muscular dystrophy disorder, Duchenne's muscular dystrophy, and that can be treated by modulating splicing, and we've discussed that mechanism in a previous slide. As I said, the 5 remaining antisense oligos function by mRNA degradation, and then all 8 siRNAs, of course, function by RNA interference by leading to the degradation of mRNA. And one thing that's interesting is that 2 of the antisense oligos that function by mRNA degradation and 3 of the siRNAs that function by RNA interference target the same gene, TTR, which encodes transthyretin. And so this illustrates a point that in some cases, for some diseases, either an antisense oligo or an siRNA can be effective strategies for gene silencing. So this raises a good question. When should you choose an antisense oligo, and when would you choose an siRNA? And so to kind of, help make that decision, I put together this table. So this shows the differences and similarities between antisense oligos and siRNA. So antisense oligos are single-stranded DNAs or RNAs or gapmers. We discussed that earlier. It has a DNA core flanked by RNA. siRNAs are always double-stranded RNAs. Both antisense oligos and siRNAs target RNA. Antisense oligos have 3 different mechanisms: steric hindrance, basically blocking translation, RNA degradation, and modulating splicing, whereas siRNA only functions by RNA degradation. Antisense oligos have 11 approved drugs. siRNA have 8, but are quickly catching up, as was noted. So the advantages of antisense oligos include, they have a variety of mechanisms. They can function by splice modulation and steric hindrance. siRNA only functions by RNA degradation. So if there's a particular disease that requires splicing modulation or steric hindrance, antisense oligos are the only choice. However, in instances where a particular protein is hyperactive and you want to downregulated by, say, inhibiting translation, degrading the messenger RNA. Both antisense oligos and siRNAs will work. Antisense oligos has the advantage that they tend to have fewer off-target effects, being a single-stranded molecule, and they're also easier to deliver. However, siRNA has more efficient downregulation, and this is probably one of the major reasons why it's been so popular, in that when trying to treat a disease, really the important thing is how efficiently you can downregulate it. So in most cases, when you're looking to downregulate a target, siRNA is probably the better choice. However, antisense oligos certainly will work in those instances and can possibly serve as a backup in situations where siRNA, that particular target, isn't amenable to siRNA for one reason or the other. However, if the mechanism requires splice modulation, in that case, antisense oligos are the only choice. Okay, now this takes us to our next section, looking at a typical workflow for nucleic acid drug development. In particular, we'll look at design, stability, as well as delivery. So let's walk through a typical workflow for developing a small nucleic acid drug, and we'll use siRNA as an example just to simplify explaining a typical workflow. So I've divided the process into three main stages. There's discovery, preclinical development, and manufacturing. So starting with discovery, the first step is to identify what protein you want to target. If you're studying a particular disease, of course, you want that protein to have a critical role in that particular disease. So once you've identified the target, the next step is to design the siRNAs to target the messenger RNA encoding that particular protein. Software tools such as GenScript's AI-powered design tools can generate multiple candidates, of which you'll need to review those to select the ones to move to the next step, which is delivery. So delivery depends on, are you going to be working in vitro or in vivo? In vitro can use standard transfection techniques. Moving into in vivo requires some more sophisticated delivery methods, such as conjugating your siRNA to a molecule to help target a particular cell type, or to use lipid nanoparticles to encapsulate your siRNA. And we'll look at both of these in more detail. So finally, when you've got the design, you've got the siRNA synthesized, and the delivery method, the next step is to then move into screening. And so you would perform in vitro screening, usually looking at RNA interference at the mRNA level. Better yet is to look at it at the protein level. And if there's any functional assays, such as maybe an enzymatic assay, those could be really useful to apply at the screening level. At the end of the discovery stage, ideally, you'd have some top candidates, possibly, usually, one candidate, but possibly more, that you would then bring into the next stage, which is preclinical development. Preclinical development includes modifications to improve the stability of your siRNA. We'll look at that in more detail. And this can be coupled nicely with an in vitro stability testing. We offered that at GenScript. It's a really fast way to quickly test those modifications, identify a modified form of the siRNA that can now be moved into your in vivo preclinical models. And then following the preclinical development, ideally you'd have one top candidate. It's also possible that you might have a backup candidate, which you now move into scaling up towards GMP level production in order to make enough material and high quality material to support an IND submission, and then clinical trials, the 3-phase clinical trials, and then ideally getting a biological license agreement and marketing your product at the end. Keep in mind, this workflow is really focused on therapeutic development, but of course, siRNAs, antisense oligos, even aptamers can be used for research applications. So a lot of this applies. In the case of research, you probably wouldn't necessarily continue into preclinical development, and you certainly want to go to GMP manufacturing for a molecule that's only going to be used as a research tool. So at GenScript, we offer a one-stop solution for nucleic acid drug development. So what this means is that we offer all of the services all the way from design, synthesis, delivery, and ultimately screening under one roof to make it really convenient, efficient and fast. So once a target is identified, we offer AI-powered design tools that can help customers design both siRNAs as well as antisense oligos. And we've newly released a tool to help design modifications for the preclinical development of siRNA to help improve stability. We also include some proprietary modifications. We'll be looking at that in our case studies. As far as synthesis, we have high throughput capabilities. We have a flexible scale all the way from nanomole up to kilograms. We also have as fast as a 4-day turnaround time for 5 nanomolar of siRNA. We have HPLC purification. We have a range of QC, including custom QC. Our standard is mass spec HPLC. Finally, as far as delivery options, we do provide LNP lipid nanoparticle encapsulation. We can conjugate siRNAs to a range of molecules, such as GalNAc for delivery to the liver. We'll look at that. We can also do antibody and peptide conjugation. And then finally, we offer a suite of screening of assays and various cell types and cell lines. So we can do in vitro screening of siRNA and antisense oligos, as well as customized functional assays. So basically, starting all the way from the target and finishing to a fully validated siRNA or ASO, ready to go into a client's experiments. To make things even easier, we've bundled our delivery services into 3 convenient packages. So starting at basic, we will design and synthesize antisense oligos or siRNAs against a customer's target and perform 1 round of in vitro screening. Our economy package adds a 2nd round of screening where we can now test for IC50 of candidate siRNAs or antisense oligos. And then our premium package adds modifications to siRNAs or antisense oligos, and those are followed by 2 additional rounds of those modified of the oligonucleotides. In addition, we also offer functional assays such as off-target analysis and stability assays. Turnaround time is really fast. You can see 6 weeks for basic, 8 weeks for economy, 12 weeks for our premium. And this allows customers to focus on other things rather than the design and synthesis and initial testing. So just leave all of that work to us. Okay, so let's look at the individual steps of nucleic acid drug development. And we'll start with the design of an siRNA. So as mentioned, GenScript has very powerful AI-based software tools to design siRNA, so you can leave a lot of the work to us. Clients only need to know what target species they're working in, what's the gene that they want to target for knockdown, if we're talking about an siRNA, and then what is the particular transcript. They need to know the transcript to knockdown. And sometimes this can be a little bit challenging. So in many cases, clients just know that they want to knock down a particular gene or particular protein. In this cases, we would recommend just knocking down, so targeting the canonical or the predominant isoform, and that's represented here. So here's a typical gene, and there's 5 predicted transcripts. Let's assume that the top one is the canonical, the predominant form. So in most cases, we would recommend, using this form as input for the software tools. The software tool would then identify siRNAs against different parts of the transcript. And so if you want to knock down every known transcript, just simply select an siRNA that recognizes a conserved region of the transcript or the gene. And that's represented by siRNA1. You can see it recognizes exon 1, which is present in all of these predicted transcripts. The flip side of that is if you want to only knock down a specific transcript, so say in this case you only want to knock down transcript number 4, in that case, choose the siRNAs that are designed that are specific for a sequence that's unique to that transcript. So that would be represented by siRNA number 4 here. So after choosing your sequences based on whether you want to knock down a single transcript or all of them, then the final step is just to review those sequences. Most of these tools, including our own, report on and off target score. So on target score is how well it recognizes its target sequence, and off target score is predicted nonspecific binding. So you want high on target binding. and poor off-target binding, or really, in other words, you want your sequences to be specific only to your target. So one of the major challenges in developing nucleic acid drugs is that they tend to be very unstable, particularly RNA, as it's sensitive to RNases, which are really found everywhere. So to help overcome this issue, small nucleic acid drugs are usually modified, and this reduces their sensitivity to enzymes such in degradation, such as RNases. Some of the common modifications to help stabilize nucleic acids are shown here, and these include modifying the phosphate backbone, the ribose sugars, as well as using modified bases. So this figure shows as reference unmodified RNA or unmodified DNA. One of the common modifications is to replace the phosphate backbone with a phosphorothioate. In addition, the ribose sugars can also be replaced with modified versions. This shows two of the common ones, 2'-O-methyl or 2'-fluoro. And then finally, the bases themselves can be modified. So in this case, cytosine could be replaced with 5-methylcytosine, or uridine could be replaced with either uridine or pseudouridine. And in both cases, these modified bases still retain function of the natural bases, but are less susceptible to degradation. Finally, another type of modification can include things like GalNAc addition. It doesn't necessarily stabilize the siRNA, although it can stabilize the end that is attached to. But more importantly, GalNAc conjugation can target siRNAs for delivery to the liver. So if the siRNA or antisense oligo needs to function in the liver, adding GalNAc is an excellent way to get it there. And just shown here is in actual real-life siRNA vutrisiran. We discussed this. This is one of the approved drugs. And as you can see, every single base is modified, either a 2'-O-methyl represented by the lowercase m or a 2'-fluoro represented by the lowercase f. In addition, there is a phosphorothioate backbone in a portion of both the sense and antisense strands, and trigalNAc is conjugated to the 3' end for liver targeting. So GalNAc conjugation isn't the only way to deliver siRNAs. So in vitro, small nucleic acid drugs can be delivered to the cell using standard transfection techniques. That's represented on the left side here. So if this is in vivo or ex vivo, in the case when cells are removed from a patient, manipulated in culture, and then returned to the patient. Small nucleic acids can be delivered by standard transfection methods, either electroporation or a lipid or chemical-based transfection. However, these methods, of course, don't work in vivo, and additional methods need to be developed. And one of the biggest breakthroughs was the development of lipid nanoparticles, as shown here. So lipid nanoparticle, it's a lipid vesicle, has a single lipid membrane, and it is surrounding a dense core, and in the dense core contains the payload, in this case siRNA, but can also contain antisense oligos, and these can be efficiently delivered into particular cells in the body. They not only protect the payload during transport, but what happens is these lipid nanoparticles are endocytosed by cells very efficiently. Once in the cell, the payload is released by endosomal escape, where they are now in the cytoplasm and they can now function. So the development of lipid nanoparticles was a huge advancement in particularly the development of siRNA. As I mentioned, another way to transport siRNAs or antisense oligos is by GalNAc conjugation. And furthermore, other conjugation methods are being explored, such as conjugating to peptides, as well as antibody conjugation. So other delivery methods are being developed that really target specific cell types, and one of those types, one of these technologies is targeted LNPs. It's something that we've been developing at GenScript, and so this shows a couple examples of our targeted LNPs. There's two main ways to do this. The first is to actually attach a ligand to the LNP. We've attached an anti-CD3 antibody that can target LNPs to T cells, an anti-CD34 antibody to target to hematopoietic stem cells, and trigalNAc itself can actually be conjugated directly to the LNP rather than to the siRNA itself to target liver. In addition, LNPs can have different formulations. So LNPs are made up of four components, four lipid components, and different lipids or different combinations of lipids can be used, and those result in targeting LNPs for different cell types or different tissues such as liver, spleen, and lung. And shown in the right side is biodistribution in mice after 24 hours. So you can see GalNAc attachment. Now the entire LNP, or most of the LNP, goes to the liver, whereas for unconjugated, you get about 2/3 going to the liver and 1/3 going to spleen. For our spleen targeting LNPs, you see that now over 80% goes to the spleen. Similarly, for our lung target LNPs, more than 85% goes to the lung. So this is the latest development, and it's definitely going to develop further to include additional cells and tissues, but it's a really powerful way of getting your small nucleic acids drug to the particular cell type that you need to get it in order to function properly, in order to hit its target. Okay, now we are going to round up today's webinar looking at some case studies that illustrate the principles that we have been discussing. So in this first case study, we designed, synthesized, and tested 150 different antisense oligos and determined the knockdown at the mRNA level, so looking at percent knockdown. Shown is a known positive control in antisense oligo that's been previously shown to efficiently knock down its target. So among the 150 candidates, you see that we were able to identify 9 antisense oligos that had not only better knockdown than the positive control, but in each case greater than 90% knockdown, which is pretty phenomenal for RNA interference. Now, case number 2 is very similar, but now we're looking at siRNAs. In this case, we only needed to design and synthesize 50 siRNAs. And as you can see, out of the 50, 13 perform a positive control. So this is a positive control for siRNA. So these two figures really illustrates 2 things. First of all, that you can see that siRNA is more efficient than antisense oligos. We needed to screen fewer siRNAs compared to antisense oligos to identify those that have very efficient knockdown. And furthermore, you can see that not every sequence that is designed is equal. So if you only design one sequence and tested it, you could have been unlucky and found one that only knocks down about 50%. So this really illustrates the importance of designing and testing multiple candidates. And this point is illustrated on the next slide. So in this slide, at the top, we're looking at 2 different genes, TTR, transthyretin, as well as NRAS. In both cases, we've designed and tested 3 siRNAs against the same target. And you can see on the left side, 2 of the siRNAs worked really well, got over 80% knockdown. One worked less well. It still got 75% knockdown, but in this case, this really illustrates why you want to test multiple siRNAs, because you want to find the one that works the best and that's most efficient. The right side illustrates a lucky case where all 3 siRNAs function equally well and all knock down greater than 90%. So on the bottom part of this figure, we're looking at 2 additional genes. In one case, we have 3 siRNAs, and in the other case, we only have 2 siRNAs. But these data basically show a comparison of GenScript's design as well as synthesis compared to siRNAs designed and synthesized by other suppliers. And as you can see, ours outperformed the other suppliers that we've tested. So to kind of summarize the results, in every gene that we've designed siRNAs for, in this particular experiment, we see knockdown efficiency over 80%, across all 4 different tested genes. We see GenScript does outperform other suppliers, and this also illustrates the importance of needing to screen more than one siRNA. And so accordingly, we have now an siRNA guaranteed package, so we can design and synthesize 3 siRNA targets against human, mouse, and rat genes, and we guarantee that at least one is will have greater than 80% knockdown at the messenger RNA level. We also offer positive and negative controls, and we have both a standard and premium package. Standard package is just the siRNAs. Premium includes the positive and negative controls. Okay, so moving on to case study number 4. So this illustrates that our knockdown isn't just at the mRNA level, that it is also at the protein level. In this particular experiment, once again, we have 3 siRNAs, each against 2 genes. And instead of looking at just the mRNA level, we look at the protein level with the Western blot. And in both cases, you can see that 2 out of 3 of the siRNAs give a really good knockdown at the protein level, almost 100%. And so this illustrates that our siRNAs not only work at the mRNA level, but they also work very effectively at the protein level. And ultimately, you probably want, in most cases, want to knock down at the protein level. So our 5th case study is looking at our stability test. So we have an in vitro stability test that we offer, and this shows that test in action, and it also shows the effects of modification, this in the next case study. So in this study, we took givosiran, one of the approved drugs. We have synthesized it without GalNAc, without a 3' GalNAc, and with. We've incubated it and took time points, and as you can see, starting at 0, where we have 100% of the starting material, as the stability assay continues to run after 4 hours, we can see that we have significant degradation of the antisense. Almost half of the antisense molecules have some sort of degradation. Not so much on the sense. When we do mass spec analysis, we can see that in the case of the antisense, we're losing the last nucleotide or the last 2 nucleotides of those molecules. When we attach GalNAc to the 3' end of the antisense, we now no longer see that degradation. So GalNAc addition not only is targeting givosiran for transport to liver, but is also helping to stabilize that 3' end. And the importance of modifications is really illustrated in a similar study. So once again, this uses GenScript's in vitro stability assay. We've incubated an antisense and a sense strand, so an siRNA duplex, either unmodified, shown here, as well as with 6 different modifications. And you can see the unmodified is pretty much degraded after 3 hours. And different modifications, these are proprietary modifications, offer increasing levels of protection for this particular siRNA, and especially our modifications number 6. You can see that after 3 hours, the siRNA is more than half intact. So this really illustrates the power of our in vitro stability. You can see how quickly you can test for stability, as well as illustrating the how well modifications can help improve stability. Okay. So now let's just summarize some of the key take-home messages that we've presented during today's webinar. So first of all, what are small nucleic acid drugs? Well, I hope, if nothing else, you've learned that small nucleic acid drugs are small oligonucleotides, RNA or DNA, that are 15 to 60 base pairs in size. They can be used to treat disease. They can also be used for research applications as well. Small nucleic acid drugs include antisense oligos, siRNAs, and aptamers. We haven't spoken too much about aptamers in this webinar. They're a little bit more complex as they fold into 3-dimensional structures that need to recognize proteins, but we've certainly discussed antisense, oligos, and siRNAs and their power for silencing genes and downregulating protein expression. To date, nearly 20 small nucleic acid drugs have been approved for human therapeutic use, many in the last several years, which really demonstrates that this is an active field of development, and we can expect to see a lot more therapeutics being developed in the oncoming years. The critical steps of nucleic acid development include sequence design, modifications, and delivery, and GenScript can help you with all 3 of these. This takes us to the last point. GenScript offers a one-stop solution, so we can do everything under one roof from design to synthesis, and we can do it as fast as 4 business days. So thank you very much for joining us in today's webinar. I hope you found it very interesting, informative, and I hope you got some insight into this fascinating world of small nucleic acid drug development. And now we will continue to our question and answer session.
Well, first, I want to say thank you very much, Mark, for that very insightful presentation. I hope everyone enjoyed it. And now, before we get into the Q&A portion of the webinar, Mark, I have 2 poll questions I'm going to launch. So let's launch this first one here. And our audience members should see it appear on your screen. Now, this you have here, you can select all that apply. This is done in real time, so your participation is strongly encouraged and very much appreciated. I'll leave it open for about 45 seconds. And the question we have for you is, what are the key factors in choosing a qualified partner? So please go ahead and make your selection, and you can choose more than one. So choose all that apply. Okay, that looks like it is half of our audience. If I can encourage the rest of our audience now to participate in this poll question, very simple poll question for you. Just click on any of the options below, and then I will close the polls, take a look at the results, and then we'll launch the second poll and take a look at the results there, and then we'll get into the Q&A. So as a reminder to our audience members, you still have the opportunity to send in your questions right now using that questions window. So please go ahead and you can also do that too if you've already done with your selection here with this poll question. Okay, that looks like it's the majority of our audience, so thank you very much, everyone. Let's take a look at the results. Okay, Mark, in our audience, 88% said innovative drug discovery and design precision, then 25% said unique modifications, then 63% all the way through to drug delivery methods, screening platform, the disease model functional assays, and then, of course, cost and delivery. All had 63% of our audience choosing that. Okay, now I'm going to go to poll question number 2. And there you go, everyone. Just like we did with the first poll, it's done in real time, so your participation is strongly encouraged. And this selection here is only one, so select one of the below. Would you design the siRNA or ASO sequence yourself, or use a third-party company for better accuracy? Okay, that looks like it is the majority of our audience. I'm now going to close the polls. Let's take a look at the results. 100% said use a third-party company. So thank you very much for sharing. Before we get into the Q&A, Mark, any thoughts on the results of that poll question 1 or 2? Anything surprising there?
Yeah, actually, that second one I was really surprised that was 100% use a third-party company. So that's really encouraging for GenScript because we do offer a really powerful software tool, AI-based software tool. So yeah, this provides a lot of insight for us into what our customers are looking for, and that they really are looking for support with designing. So it's really exciting.
Yes, it is. Okay, so thank you very much, everyone, for participating in the poll question. So just a reminder again, we are now going to begin the Q&A portion of the webinar. So if you are attending via GoToWebinar, please use the questions window. And for those of you watching on LinkedIn Live events, feel free to post your comments right now in the questions section. Okay, now let's get into the Q&A. Now, Mark, let's start with this question here. This is, let's see. Oh, this audience member is asking, can siRNA target small RNAs such as miRNAs? A great question there, Mark. What do you think?
Yeah, that is a very good question. Usually not, just because really small RNAs just are too small to find a good target sequence. So in this case, if you want to target a small RNA like a microRNA, probably an antisense oligo is your best approach, because just the target sequence for siRNAs is too limited on such a small molecule.
Okay, thank you very much for that, Mark. Let's jump to the next question here. This audience member is asking, how long does siRNA knockdown last?
Oh, that's a good question. I mean, it really depends on a lot of factors: how stable the RNA is itself, what you're targeting, the half-life of your protein, the half-life of the mRNA. But it can be anywhere from a day to possibly as long as a week or more. So it really depends on a lot of factors. And so I would encourage anyone working on siRNAs is to really kind of do close analysis of their particular targets, the siRNAs that they're working on, both in vitro and in vivo models.
Okay, perfect. Thank you so much. Let's jump to this next question here. How long after transfection with an siRNA should I screen for knockdown?
Oh, that's a good one. So that's really related to the previous question. So once again, it depends on the stability, the half-life of the protein that's being encoded, the messenger RNA or the RNA that you're targeting. But I would say in general, you could start looking as early as 24 hours, but certainly looking anywhere from 48 to 72 hours. If you're going to see an effect, you would probably see it in that range. If not, I would, if you're looking at 48 or 72 hours and you don't see any knockdown, I would encourage you to maybe look at 24 hours and then maybe look a little bit longer beyond 72 hours. I would imagine, anything past 4 days or so, if you don't see any effect by that point, unfortunately, you probably won't.
Okay. All right. Thank you for that. Looks like we have 2 more questions here. So just as a reminder to our audience, you can still send in your questions now using that questions window. Now let's jump to this next question here. This audience member is asking, are there any small RNA therapies used so as to treat animal diseases? That's an interesting question, Mark. Yeah, what do you think?
Yeah, I imagine that's to treat veterinary and livestock diseases. That's a really good question. We could probably have a whole webinar on that. As of now, I'm not aware of any. So I'm not aware of any approved therapeutics, siRNA therapeutics to treat animal diseases. But I know a lot are in development, and you can imagine, different applications, a lot of equine diseases, horse diseases. Also, with avian flu, using siRNA or even other RNA-based approaches could probably be very effective. So it's definitely an area of active development. But to date, as far as I know, there aren't any approved therapeutics for veterinary use.
Okay, thank you so much, Mark. This looks like it's our last question here. This audience member is asking, how predictive is the biodistribution and knockdown in a mouse study using an LNP for targeting specific organs and cells in human?
Oh, that's another great question. Because when you run a mouse study, you definitely want it to be applicable to treating human diseases. So, with a lot of the mouse studies with the targeted LNPs and even other targeted ways, a lot of those have not been looked at in humans just because it's very invasive to look at, biodistribution in humans, particularly going to certain organs. But there's a lot of evidence that supports that the mouse studies are translatable directly to humans. So first of all, GalNAc conjugation. In that case, we know that GalNAc conjugation can certainly target liver diseases very effectively. So presumably, in those cases, GalNAc is delivering LNPs as well as siRNAs to human liver. So in that case, it is very the mouse studies are very predictive of human diseases. For some of the other tissues, we haven't, it hasn't yet been looked at. I should say to look at human biodistribution, the approach that's taken is to go in vitro, to look at different cell types in vitro from different organs or expressing different markers, things like, T cells, certainly in vitro. We know that LNPs conjugated with an anti-CD3 antibody to target T cells will work, in vitro. And a lot of the T cell work, of course, a lot of the therapeutics is done in vitro, ex vivo, for development of therapeutics such as CAR. So, just because of the invasiveness of looking at these studies in humans, we haven't been able to generate that data, but it's very likely that mouse studies are going to be highly predictive to human biodistribution just based on what we've looked at so far with GalNAc and things like T cell targeting. So yeah, thank you for that question. That's a really good question.
Yes, thank you very much for that detailed answer, Mark. Well, it looks like we reached the end of the Q&A portion of the webinar. So I just want to say for those of you who are interested in hearing more from GenScript, you can register for their next webinar, Unlocking the Future of Gene Editing, Key Concepts and Breakthroughs in CRISPR Prime Editing with Juliana Campo Garcia, the Field Application Scientist for GenScript. And that is scheduled for May the 9th. And I'm going to put the link in the chat box here so that it makes it easy for you to go ahead and register for this next exciting webinar that we have planned here for you. Okay, now, if you did not get your questions answered or if you have any questions, further questions or resources you're looking for, please utilize the email address you see there on your screen, and there's one there just for Mark. Okay, all right. Now, Thank you very much, everyone, for participating in today's webinar. You will be receiving a follow-up email from XTalks with access to the recorded archive of this event. A survey window will be popping up on your screen. Your participation is appreciated as it will help us to improve on our further webinars. Additionally, give me a few seconds. I'm going to share the link of today's recording event in the chat box, which you can also share with your colleagues once they register for the recording here as well. And for those of you watching via LinkedIn Live events, we encourage you to visit xtalks.com to register for this event and gain access to features such as viewing the recording and more. Now, please join us in thanking our speaker for his time here today. So thank you very much, Mark, for your time. It was great listening to you. We hope you found this webinar informative. It has been my pleasure to be your webinar moderator. On behalf of the team here at XTalks, we thank you for joining us. Until we meet again, I'm Sonia Hunt. Please take care and bye for now. Bye, everyone. Bye, Mark. Bye.